And so I live in Tokyo. It's a center of Japan. It's a capital. It's a basic information of Japan. And so now, I'm a professor of the Institute for Solid State Physics at the University of Tokyo. And so today, I'd like to talk about KIF1A from the viewpoint of physics. Almost all of the people belong to biology area in this conference, but I belong to physics department. And what is Japan famous for? Probably you know sushi, and anime is also very famous. And recently, baseball. But it is famous. The Japan is famous for motor protein studies. And so I hope in the future you have a lot of opportunity to meet more Japanese researchers, protein motor researchers. So axonal transport materials synthesized in the cell body must be delivered to synapses. The active transport by motor proteins is needed. The kinesin 3 motor KIF1A transport synaptic materials. So there are many drivers in cells. We have about 40 motor proteins. Each one have different shapes. Then you can easily imagine, the defects, there are defects of drivers. The defects of drivers are related to disease. Because materials such as protein and molecules needed in cells can't be delivered if there is a deficit of motor proteins. So related to physical quantities, characterizing ability of axonal transport. So first one is force. Because cargo, a synaptic cargo is very large, motor protein had to produce a lot of force and velocity. So if a motor protein carry a big cargo, synaptic materials, motor protein had better to run fast. So people can understand force and velocities. So they are very important physical quantity. It's very easy to understand, I think. But today I'd like to add one more physical quantity to focus the number of motors, number of KIF1As, attached to a cargo, carry a single cargo cooperatively. So we really focus on. So look at the pictures. This is a portable shrine. This is a portable shrine. So we usually carry a big portable shrine together with many people. I think these kind of the many people, multiple kinesin transport make efficient transport. So number of kinesin molecules attached to a cargo, why is it important? So the answer is it alters the run length of the transport. So detachment occurs stochastically, so it is called run length. Sometimes it's KIF1A, at first, KIF1A run along the microtubule, it detaches from the rail stochastically. It is called run length. But imagine if there are many kinesins at the same time carry one cargo, so the run length becomes longer because somebody is attached to the rail in the case of multiple motors. Indeed, it was reported the run length affected a lot to synapse formation and velocity. I can't read this part. I can't read this part. I forgot what. Right. The ability to, the ability to continuously transport materials, very important. So then, what is the appropriate number of kinesins, KIF1As? So this is our hypothesis. So there is appropriate place, positions for synapse creation. The KIF1A should unload cargoes at that place. But however, so if there is a KIF1A cooperatively, so transporter cargo is, so if there is a small number, so it detaches very soon. So if so, thank you very much. If so, I can erase this part. It's easy for me to read. If so, there are a lot of kinesins. So run length becomes very long. So probably that if the number of kinesin is small, or if the number of kinesin too many, probably it is difficult for them to unload cargoes at the appropriate place. Then the mislocation for synapse creation occurs. Synaptic material transport by appropriate number of KIF1A seems important. So how to measure the number of kinesin molecules? So super resolution microscopy may not work well for the counting. So it may not distinguish active and inactive motors. So on the cargo membrane, there are a lot of KIF1A molecules, but some of them are inactive motors, not participating in the transport. So active motors transporting the cargo is, so in this schematics, there are 3. So our strategy is to develop a software rather than microscope. So that we suggest a new physical quantity chi to count the number of active motors, active kinesins. So we believe physics is a powerful tool. So let's see the result of the observation of synaptic cargo transport in C. elegans. So by the way, it's a worm. You can see the worm is moving. So it's a small worm, but its neurons have a lot of similarity to ours. Inside this worm, you can see a cargo moving, transported by KIF1A. Now, I think you can see it from here to there. This is another movie. In this way, even a worm is living, so it's possible for us to observe the cargoes moving, transported by KIF1A. And using a self-made tracking software, we can track single cargoes. Then we measure the chi to count the number of KIF1As. So here I don't plan to talk about the physics lecture, but just I'd like to tell a point. So usually people measure the velocity, but in our case at the same time we focus on the fluctuating behavior. So you can see this is the cargo position as a function of time. So you can see a fluctuating behavior. It's not straight, but it fluctuates. And this fluctuation has information based on the physics. And we combine both velocity and fluctuation, and then obtain the information of number of active kinesins. So you don't have to follow them now. But the information how to calculate, how to connect the number of active kinesin to that measurable quantity chi is in this review. So anyway, the point is we can count the number of KIF1A transporting the synaptic cargo. The synaptic cargo was transported by about 1 to 4 kinesin, so KIF1A molecules. So measuring the mutant, C. elegans, measuring the chi for mutant C. elegans. So it was reported that the mutant C. elegans caused the mislocation of synapses. So look at the pictures. Usually, this is the cell body, and the synapse creation occurs. This is a synaptic area, but in the case of mutant, So in this case, the synapse is created so close to the cell body. It's a mislocation of the synapses. So using this mutant, so in this mutant, so the lack of accessory protein, so axonal protein, so called R8, decrease the number of KIF1A. I mean that usually KIF1A takes an inactive form. So when it doesn't attach to a cargo in order not to use energy too much. And then this R8 protein usually help to release in auto-inhibited form of KIF1A to active form. So if there is no R8, there are few the number of active kinases decreases. So the point is, using that chi, we can measure the decrease of the number of kinesins, KIF1A. In this case, 20% decrease in the number of active KIF1A molecules, in this case of the mutant worms. So finally, the measurement of chi for disease model, C. elegans. So hereditary spastic paraplegia model, C. elegans, was first created in the previous study and known as KIF1A, associated neurological disorders. The point is, in the case of wild type, the worms, this worm moves very fast, but in the case of mutant worm, this motion becomes very slow. And what we found is, in the case of this mutant worm, we can see the number of kinesins increases this time. So excess transport by many active KIF1A might occur in this mutant. So as I told you, there is an appropriate place for synapse position, and the appropriate the number of kinesin KIF1A shouldn't be too small, but at the same time shouldn't be too large. The appropriate number of KIF1A seems important for healthy axonal transport. So finally, in order to understand the quantitative understanding of synapse creation, we try to construct a model. So in order to connect the number of kinesin to synapse creation, recently we start to creating a simulation model. So many cargo are moving, and you can see some cargo stops. Yeah, some cargo stops, and it's the origin of synapses, we think. So towards the understanding of human neurons, so we recently started the culture of iPSC motor neurons. That we successfully, so we can see that on the motion of synaptic vesicle precursors that transported by KIF1A. So we got the preliminary data. The result of chi measurements are coming soon. So finally, the epilepsy. So my coworker, in fact, passed away 2 years ago. And I already think what I can do for neuroscience area from the physical point of view. And I hope our technique may help to understand the mechanism of CAND in the future. Thank you very much for your attention.
very interesting. Thank you for the talk. I just had a question about the physics model. To my understanding, you said that you track the vesicles, right, or the cargo, and then you can be able to calculate the number of kinesins moving that cargo. So I was wondering if this model takes into account, you know, like, can it differentiate which kinesin is actively on that vesicle? Because like even for synaptic vesicle precursors, there's different kinesins like KIF1B beta, I believe even kinesin 1. So can you differentiate between those specific motor classes?
Probably so that if so there are the stall force of the motor is different, probably so that we can differentiate. But so in the case of KIF1 and KIF5, the stall force is very similar. It's very difficult for us to distinguish. So the value of stall force, using the value of stall force, we can distinguish them. But I think so they are very similar.
Okay.
If the force is quite different, so that we can so we can differentiate two motors.
Force.
And depending on the force value.
Thank you for the talk. That was really interesting. You said that in the mutated version, you can see formation of synapses at the wrong places. Can you see something that would mimic an epileptic seizure in this way? Do the worms have something resembling that? And could that be a cause for epilepsy?
It's very difficult still now, so that still so that we don't understand why it is the location becomes abnormal. Still we don't know the mechanism, so I can't tell in the case of epilepsy. So still so that I don't understand in the case of CAND, we don't understand. So even more in the epilepsy, in the case of epilepsy, so no idea. But that's the reason why we started so creating the model to understand it. I hope so that both CAND and epilepsy in the future, the position of a cargo is predicted from the mutation of so KIF1A or so the mutation of kinesins. The mutation of axonal transport, then so that we hope to predict the position of the synapse in the future.
Can we go back to the V8M data that you presented? I just wanted to make sure. So here, are you looking at the number of active kinesins transporting, and here you're seeing that there's obviously more kinesins or for the mutant, right? So then this is a question of, is it just this mutant is being recruited more? And can you explain like maybe the mechanism of the nature of this mutation for it to want to?
So this is already so the investigated in the previous paper.
Okay.
So please check this previous paper, please. And that the fact is so number of active kinesins increases. The total amount of kinesin the same, but so active kinesin increases in this mutant according to the previous paper.
Okay.